Namaste everyone. So in the last video we have come across with the different types of inheritance patterns, and then we have come across with some other factors of the sex linkage. So in this class today we are going to determine the sex of the different organisms, how we can determine the sexes. So usually we can go through the morphological character. If they are showing the different morphological character based on the sexual dimorphism, so we will differentiate, but it is not so easy to determine such organisms which are showing the same type of the characters. It is not easily observable morphologically. So that's why there are some experiments taken place through the cytological content. So the cytological observation that lead to the genetical sex determination or chromosomal sex determination. Then what is this sex determination? Means sex determination is the establishment of the sex through differential development in an individual at an early stage of life. So ಹಾಗಾದರೆ ಸೆಕ್ಸ್ ಡಿಟರ್ಮಿನೇಷನ್ ಅಂದರೆ ಏನಂತ ಹೇಳ್ತಾ ಇದ್ದೀವಿ? ಇಟ್ ಈಸ್ ದ ಎಸ್ಟಾಬ್ಲಿಷ್ಮೆಂಟ್ ಆಫ್ ಸೆಕ್ಸ್ ಥ್ರೂ ದ ಡಿಫ್ರೆನ್ಷಿಯಲ್ ಡೆವಲಪ್ಮೆಂಟ್ ಇನ್ ಆ್ಯನ್ ಇಂಡಿವಿಜುವಲ್ ಅಟ್ ಆನ್ ಅರ್ಲಿ ಸ್ಟೇಜ್ ಆಫ್ ಲೈಫ್. ಸೊ ಅರ್ಲಿ ಸ್ಟೇಜಲ್ಲೇ ಪ್ರತಿಯೊಂದು ಇಂಡಿವಿಜುವಲಲ್ಲೂ ಸೆಕ್ಸುವಲ್ ಡಿಫ್ರೆನ್ಸಿಯೇಷನನ್ನು ನಾವು ನೋಡ್ತಕ್ಕಂಥದ್ದನ್ನ ಸೆಕ್ಸ್ ಡಿಟರ್ಮಿನೇಷನ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಹಾಗಾದ್ರೆ ಸೆಕ್ಸ್ ಡಿಟರ್ಮಿನೇಷನ್ ಅಂದರೆ ಅಷ್ಟು ಈಸಿಯಾಗಿ ಗೊತ್ತಾಗ್ಬಿಡುತ್ತಾ? ಅಷ್ಟು ಈಸಿಯಾಗಿ ಸೈಟಲಾಜಿಕಲ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಬೇಸ್ ಮೇಲೆ ಅವ್ರು ಕಂಡುಹಿಡ್ಬಿಟ್ರಾ ಅಂದರೆ ನೋ, ಇಟ್ ಈಸ್ ವೆರಿ ನಾಟ್ ಸೋ ಈಸಿ. ಇಟ್ ಈಸ್ ಅ difficult job they did. So cytological observation that lead to the development of genetical and chromosomal basis of sex determination was carried out through the first by the Henking. So Henking is the person who in 1891 could trace a specific nuclear structure all through the spermatogenesis in a few insects. So Henking ಏನು ಮಾಡ್ತಾನೆ ಅಂದರೆ ಒಂದಿಷ್ಟು ಇನ್ಸೆಕ್ಟ್ಸ್ನ ತಗೊಂಡು ಅದರಲ್ಲಿ ಸ್ಪೆಸಿಫಿಕ್ ನ್ಯೂಕ್ಲಿಯರ್ ಸ್ಟ್ರಕ್ಚರ್ ಅಂದರೆ ನ್ಯೂಕ್ಲಿಯಸ್ ಒಳಗಡೆ ಇರ್ತಕ್ಕಂಥ ಒಂದು ಸ್ಪೆಸಿಫಿಕ್ ಸ್ಟ್ರಕ್ಚರ್ನ ಸ್ಟಡಿ ಮಾಡ್ತಾನೆ. ಅದು ಸ್ಪರ್ಮೆಟೋಜೆನೆಸಿಸಲ್ಲಿ ಯಾವ ರೀತಿ ಕ್ಯಾರಿ ಆಗುತ್ತೆ ಅನ್ನೋದು ಕೂಡ ನೋಡ್ತಾ ಹೋಗ್ತಾನೆ. ಸೊ ವಾಟ್ ಈ ಅಬ್ಸರ್ವ್ಡ್ ಅಂದರೆ 50% ಆಫ್ ಸ್ಪರ್ಮ್ಸ್, ಎಂಕಿಂಗ್ ಏನು ಮಾಡ್ದ ಅಂತ ಮಾತಾಡ್ತಾ ಇರೋದು. ಸೊ ವಾಟ್ ಈ ಅಬ್ಸರ್ವ್ಡ್ ಇನ್ ದ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ಸ್ ಮೀನ್ಸ್ 50% ಆಫ್ ಸ್ಪರ್ಮ್ಸ್ ರಿಸೀವ್ಡ್ ದಿಸ್ ಸ್ಟ್ರಕ್ಚರ್. ಅವನೇನು ನ್ಯೂಕ್ಲಿಯರ್ ಸ್ಟ್ರಕ್ಚರ್ ನೋಡಿದ್ದ ಅದು. ಸೊ the Henking observed by the experiments in the insects by tracing a specific nuclear structure all through the spermatogenesis in a few insects, where he also observed that 50% of sperms received this structure in a nuclear structure. So 50% of sperms received this the specific nuclear structure after spermatogenesis, whereas 50% of sperms didn't receive this specific nuclear structure. See, Henking ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಏನು ಮಾಡ್ತಾ ಹೋದ? Henking ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡ್ತಾ ಹೋದಾಗ ಗೊತ್ತಾಯಿತು, ಅದರಲ್ಲಿ ಬರ್ತಕ್ಕಂಥ ಪೇರೆಂಟಿಂದ ಆಸ್ಪ್ರಿಂಗಿಗೆ ಬರಬೇಕಾಗಿರ್ತಕ್ಕಂಥ ಗ್ಯಾಮೆಟ್ಸಲ್ಲಿ ಏನಾಗ್ತಾ ಇದೆ ಅಂದರೆ ಒಂದಿಷ್ಟು ಅಂದರೆ 50 ಗ್ಯಾಮೆಟ್ಸು ಕಂಪ್ಲೀಟಾಗಿ ಸ್ಪರ್ಮ್ಸ್ ರಿಸೀವ್ ಆಗಿರ್ತಕ್ಕಂಥ ಗ್ಯಾಮೆಟ್ಸ್ ಇರ್ತವೆ. ದ 50% ಆಫ್ ಸ್ಪರ್ಮ್ ಏನಾಗಿರುತ್ತೆ ಅಂದರೆ ಈ ಒಂದು ಸ್ಟ್ರಕ್ಚರ್ನ ರಿಸೀವ್ ಮಾಡಿರುತ್ತೆ. ಸ್ಪೆಸಿಫಿಕ್ ಸ್ಟ್ರಕ್ಚರ್ ಅಂತ ಏನು ಕಂಡಿಡ್ದ, ನ್ಯೂಕ್ಲಿಯರ್ ಸ್ಪೆಸಿಫಿಕ್ ಸ್ಟ್ರಕ್ಚರ್ ಅಂತ ಏನು ಹೆಂಕಿಂಗ್ ಅಬ್ಸರ್ವ್ ಮಾಡ್ದ, ಸೊ ಆ ಸ್ಪೆಸಿಫಿಕ್ ನ್ಯೂಕ್ಲಿಯರ್ ಸ್ಟ್ರಕ್ಚರನ್ನು 50% ಸ್ಪರ್ಮ್ಸ್ ರಿಸೀವ್ ಮಾಡ್ತವೆ, ಇನ್ನು 50% ಸ್ಪರ್ಮ್ಸು ರಿಸೀವ್ ಮಾಡಲ್ಲ ಅಂತ ಗೊತ್ತಾಗುತ್ತೆ. ಸೊ ಹಾಗಾದರೆ ಆ ಹೆಂಕಿಂಗ್ ಆ ಒಂದು ಸ್ಪೆಸಿಫಿಕ್ ಸ್ಟ್ರಕ್ಚರ್ಗೆ ಏನಂತ ಹೆಸರು ಕೊಡ್ತಾನೆ ಅಂದರೆ ದಟ್ ನೇಮ್ ಇಸ್ ಕಾಲ್ಡ್ ಎಕ್ಸ್ ಬಾಡಿ. ಸೊ ಇವತ್ತು ನಾವೇನು ಎಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ ಅಂತ ಕರಿತೀವಿ ಅದನ್ನು ಎಕ್ಸ್ ಬಾಡಿ ಅಂತ ನಾವು ಹೆಂಕಿಂಗ್ ಪ್ರಕಾರ ಕರಿಬೋದು. ಸೊ ಹಾಗಾದರೆ ಹೆಂಕಿಂಗ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಪ್ರಕಾರ ಏನಾಗುತ್ತೆ ಅಂದರೆ ನೋಡಿ, ಸೊ ಫೀಮೇಲ್ ಯಾವಾಗಲೂ ಕೂಡ ಎಕ್ಸ್ ಇರುತ್ತೆ, ಮೇಲ್ ಯಾವಾಗಲೂ ಕೂಡ XY ಇರುತ್ತೆ ಇನ್ ಹ್ಯೂಮನ್ ಕೇಸ್ ಅಂತ ತಗೋಣ. ಬಟ್ ಅವನು ಹೇಳ್ತಿರೋದು ಈಗ ಸ್ಪರ್ಮ್ ಪ್ರೊಡಕ್ಷನ್ ಅಂದರೆ ದ ಮೇಲ್, ಇದ್ರ ಬಗ್ಗೆ ನಾವು ಮಾತಾಡೋಣ ಈಗ. ಸೊ ಡ್ಯೂರಿಂಗ್ ಆಫ್ಟರ್ ಸ್ಪರ್ಮೆಟೋಜೆನೆಸಿಸ್ ಏನಾಗುತ್ತೆ ಅಂದರೆ ಇಟ್ ವಿಲ್ ಪ್ರೊಡ್ಯೂಸ್ ದ ಗ್ಯಾಮೆಟ್ಸ್. ಸೊ ನೋಡಿ, ಕೆಲವೊಂದು ಗ್ಯಾಮೆಟ್ಸಲ್ಲಿ ಆ ಒಂದು ಸ್ಟ್ರಕ್ಚರ್ ಇದೆ, ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿರ್ತಕ್ಕಂಥದ್ದು 2 ಗ್ಯಾಮೆಟ್ಸ್. ದೀಸ್ ಆರ್ ಎಟಿರೋ ಗ್ಯಾಮೆಟ್ಸ್. ಆದರೆ ಒಂದರಲ್ಲಿ ಎಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ ಇದೆ, ಇನ್ನೊಂದರಲ್ಲಿ ವೈ ಕ್ರೋಮೋಸೋಮ್ ಇದೆ, ಎರಡರಲ್ಲೂ ಕೂಡ ಸೇಮ್ ಇಲ್ಲ. ಸೊ 50% ಆಫ್ ದಟ್ ಸ್ಟ್ರಕ್ಚರ್ ಹ್ಯಾಸ್ ರಿಸೀವ್ಡ್. ಸೊ ಆ 50% ಆಫ್ ಸ್ಪರ್ಮ್ಸಲ್ಲಿ ಆ ಸ್ಟ್ರಕ್ಚರ್ ಇತ್ತು, ಇನ್ನು 50% ಆಫ್ ಸ್ಪರ್ಮ್ಸಲ್ಲಿ ಆ ಸ್ಟ್ರಕ್ಚರ್ ಇರಲಿಲ್ಲ ಅಂತ ಕರಿತೀವಿ. ಹಾಗಾದರೆ 50% ಆಫ್ ಸ್ಪರ್ಮ್ಸ್ ಎಲ್ಲ ಸ್ಟ್ರಕ್ಚರ್ ಇತ್ತು ಅಂದರೆ ದಟ್ ಈಸ್ ಎಕ್ಸ್ ಬಾಡಿ ಅಂತ ಕರೆದ, ಅದನ್ನು ನಾವು ಎಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ ಅಂತ ಈಗ ರೆಫರ್ ಮಾಡ್ತಾ ಇದ್ದೀವಿ. ಸೊ ದಿಸ್ ಈಸ್ ಹೌ ದ ಹ್ಯಾಂಕಿಂಗ್ gave the explanation through carrying out the different experiments on the insects to determine the sex of an insect. So based on this genetical and chromosomal sex determination, we are going to perform the different experiments and then going to identify the sex of different organisms. So in this, the sex determination, we are having the 3 different types of sex determinations based on the fertilization events that takes place in an organisms. See, Progamic, Syngamic, Epigamic. So name itself is saying that pro refers to early or before. Gamy refers to fusion or marriage. So before marriage means nothing but the early fusion, means before the fusion or before the marriage, means the progamic type of sex determination is nothing but it is a type of sex determination where the sex can be determined before the fertilization. If the sex is determined before the fertilization, it is called progamic. We can observe it in the drone in honeybee. So I will detail about this in the next content. So syngamic type of sex determination means syn means both, gamy. Syngamy means it is a fusion, means during the fusion means nothing but it is the type of sex determination. Syngamy means both will fuse, syn means both, gamy means fusing, so or wedding. So both the gametes are getting fused means that is fertilization. So syngamic type of sex determination means it is nothing but the sex determination that identified during the phase of fertilization. That's we can see in most of the plants and animals especially. And then last one is epigamic type of sex determination. Epi refers to above or after, so gamic refers to the fusion. So after fusion, if we identify the sex of an organism, then it is called epigamic type of sex determination. We can see it in the female honeybee. So just we discussed about the types of the sex determination based on the fertilization event. So let us deal about the mechanism of sex determination. So the mechanism of sex determination is based on, so we are having the 2 or 3 different types of sex determination mechanisms. One is allosomic, another one is gene balancing theory, and another one is haplodiploidy mechanisms. So now we are moving towards the allosomic mechanism of sex determination. Allosomic, as the name allo refers to the, it indicates the sex. So soma means the body. So the chromosomes which are controlling the sex characters, so they are all called allosomes, and the chromosomes which control the body characters are called autosomes. So the sex is inherited through these allosomal, means the sex chromosomes. Hence the allosome is also having the synonym called sex chromosome. It is having either X or Y or Z or W likewise. Hence they are called heterosomes. Hetero means different. So X and Y both are different. So allosomes are Z and W. They are also different. So in generally they are called as heterosomes also. So the name sex chromosome was given by Montgomery, and then that was the sex chromosomes were first discovered in grasshopper by McLeod Klung. So as we already studied, Henking gave the name called sex X body, and then this is now we are referring as a X chromosome. And so Wilson and Stevens did the experiments and they gave the theory for determining the sex called the chromosomal theory of sex determination. It is not chromosomal theory of sex inheritance. So it is a chromosomal theory of sex determination. So they have taken an insect called ligase. So that's shown the sex type called XXXY type of the sex determination. So hence that is called the ligase type of sex determination. So let us see. How? So in this XX, XY are the ligase type of sex determination. Here we can see XX female, XY male. So what happens? See, XX female with XY male. So X produces the gametes as same type of gametes after meiosis, but whereas the male produces X and Y as the gametes after the meiosis. So we can observe this. X and X both are same type, hence it is homo. These two are the gametes, hence we can call it as homogametes. Here in the male we can see X and Y are the two different, hence it is called as hetero. And then XY are the gametes which are from the male, hence heterogametes. So that's why the type of sex determination XX female and XY male can be seen as female homogamety, male heterogamety. So female producing the same type of gametes, hence it is called female homogamety. Male is producing the two different gametes, called it as heterogamety. And then here we can observe the male is having the two possibilities to give the progeny. Means here female is giving only X. If it is fused with the male gametes, we can have the two possibilities. If the male X chromosome and the female X chromosome fuses, means we will get the daughter in the progeny, means female. And if the X chromosome and the Y chromosome of the male, X chromosome of the female and Y chromosome of the male get fused, means we can get the XY as the progeny, means the son or a male progeny in the F1 generation. Then how it is happening? Means the male produces two type of sperms. One which contains the X chromosome is called gynosperm. The chromosome which contains the sperm which contains the X chromosome is called as gynosperm, and then the sperm which contains Y chromosome is called androsperm. Okay, so this is the sex determination in the human we can see. So XX female, XY male type of sex determination can be seen in human and most of the mammals, and especially in some of the dioecious plants. So this is the XX female, XY male. Here the sex determination is female homogamety, male heterogamety, and male is producing two different types of sperms. As I just said, gynosperm means the sperm which contains the X chromosome, and the sperm which contains the Y chromosome is called androsperm. So this type of XX female, XY male type of sex determination can be seen in man and dioecious plants. So and then another type of XX, XY or the ligase type of sex determination is XY female and XX male. Means as we just seen, the same pattern can be seen in most of the insects. Means the butterflies and moths. So in butterflies and moths we can see the same type of inheritance pattern. XY is female, means XY is female, means XY female crossed with XX male. But whereas in the man and dioecious plants we can see female as XX, but here the female is XY is crossed with the XX male. See, here we can expect the gametes as X and Y, and here we can expect the gametes as X and X. So female is producing two different types of gametes, whereas male is producing same kind of gametes. So as we said, if it is producing two different kind of gametes, it is called heterogamety. It is called homogametes because producing the same kinds. So female heterogamety, male homogamety condition can be seen in insects, especially butterflies and moths, and even this ligase type of sex determination can be seen by the female heterogamety, that is ZW, and the male homogamety, that is called ZZ. See what happens means, here instead of X we can see the Z chromosome, instead of Y we can see the W chromosome, but female is here ZW component and then male is having the ZZ component. So what happens when it produces the gametes, one gamete will contain in the Z chromosome, another gamete contains the W chromosome. Here in the male it produces two same kind of gametes, that is having the ZZ. So what happens means, female is producing two different gametes, one is Z, another one is W, means one gamete containing Z, another one containing the W chromosomes, hence it is also female heterogamety. But whereas the male is producing same kind, hence homogametes, the male is homogametic condition. So this ZW female, ZZ male means the heterogametic female and homogametic male condition of sex determination by the ZW and ZZ type is seen in birds and even in some of the vertebrates like the fishes and reptiles. Coming to another type of sex determination in an allosomic type of or allosomic mechanism of sex determination is XX female, XO male. See here, XX means female is crossed with XO of the male. See, either in the last cases we have seen that either male or female will be having the heterozygotic or heterogametic or homogametic, but here the male is lacking one of the chromosome. The male is lacking one of the chromosome, and see the female produces two type of same gametes, but whereas the male produces only one gamete and another gamete lacks the chromosome. So hence it is female homogamety, male heterogamety. So this type of sex determination can be seen in grasshopper and then squash bug anasa, and in cockroach, ascaris, and some plants like Vallisneria spiralis and Discoria sinuta. So this XX female and XO male is also called as proteiner type of sex determination. Here the male lacks one of the chromosome. Okay, so it produces the gametes along with the autosomes with the allosomic chromosomes. Okay, so in the male we can expect only the different kinds of set of chromosomes present in the gametes, but whereas in the female we can expect the same kind, hence it is called as the female homogamety, male heterogamety condition. So this is another type of the sex determination in an allosomic mechanism of sex determination patterns. Coming to very important and another one is the haploid diploid mechanism. As I said, there are three different mechanisms. One is the allosomic, another one is the haploid diploid, and another one is the genic theory. So now we are going to discuss about the haploid diploid mechanism, especially in the honeybees. So what happens means, in the honeybee, especially in the order, means the insects which are the order of Hymenoptera, especially ants, wasp and the honeybee, they are all comes under the order called Hymenoptera taxonomically. So here the sex determination of these insects can be done through the set of chromosomes which are containing. If they are containing the diploid set, two set of chromosome, then it will be the female If they are containing the haploid number of chromosome, one set of chromosome, then they are called as male. So here the special mechanism that takes place is parthenogenesis, because the parthenogenesis is the mechanism where the progeny is developed without fertilization. So the progeny which developed without fertilization is here the male drone, which develops from the unfertilized eggs. That's why it is haploid. And then male is always called as parthenote, because it is a progeny developed through the parthenogenesis. But queen and workers both are the developed from the fertilized eggs. So let's see how they are means. If the female honeybee is having the 36 chromosomes, that after the meiosis produces the gametes which containing 16 and 16. These two undergoes the mechanism, one which gets fused with the other female gamete, then it will form the diploid. But here which continues as it has means without fusing, without fertilization, it is through the unfertilized egg which develops into the male. See, that's why I said diploid set of chromosome leads to the female, haploid set leads to the male. So ಯಾಕೆ ಅಂತ ಹೇಳಿದೆ ಅಂದರೆ ಇದೇ ರೀಸನ್ಗೆ. Clear? So likewise in this insects, especially the order Hymenoptera, which contains the insects like ants, wasp, honeybee, we can see the haploid ಅಂದರೆ ಮೇಲ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಇರುತ್ತೆ, ಫೀಮೇಲ್ ಡಿಪ್ಲಾಯ್ಡ್ ಇರುತ್ತೆ. ಮೇಲ್ ಯಾಕೆ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಆಗಿರುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಇಲ್ಲಿ ನಾವು ನೋಡಿದ್ವಿ, ಮೇಲ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಆಗಿದೆ ಅಂದರೆ ಪಾರ್ಥೆನೋಜೆನೆಸಿಸ್ ಪ್ರೊಸೆಸ್ ಮೂಲಕ ಅದಾಗಿದೆ ಅಂತ. ಹಾಗಾಗಿ ಇಟ್ ಡೆವಲಪ್ಸ್ ಆಸ್ ಅ ಡ್ರೋನ್. ಸೊ ಆ ಒಂದು ಮೇಲ್ಗೆ ನಾವು ಡ್ರೋನ್ ಅಂತ ಕರಿತೀವಿ. ಫೀಮೇಲ್ ಏನಾಗುತ್ತೆ ಅಂದರೆ ಇಲ್ಲಿ ಕ್ವೀನ್ ಆ್ಯಂಡ್ ವರ್ಕರ್ ಬಿ ಆಗುತ್ತೆ. ಯಾಕೆ ಈ ಥರ ಆಗುತ್ತೆ ಅಂತ ಮಾತಾಡೋದಾದರೆ ಇಲ್ಲೇನಾಗುತ್ತೆ, ಫೀಮೇಲು ಯೂಶ್ವಲಿ ಕಂಟೆಂಟ್ ಏನು ತಗೊಳುತ್ತೆ ಅಂತ ನೋಡೋದಾದರೆ, ಸೊ ಲೆಟ್ಸ್ ಸಿ. ಸೊ ಫೀಮೇಲು ಫರ್ಟಿಲೈಸ್ಡ್ ಎಗ್ಗಿಂದ ಡೆವಲಪ್ ಆಗುತ್ತೆ. ಹಾಗಾಗಿ ಫರ್ಟಿಲೈಸ್ ಆದಾಗ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಆ್ಯಂಡ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಎರಡು ಸೇರಿದ್ರೆ ಡಿಪ್ಲಾಯ್ಡ್ ಆಗುತ್ತೆ. ದಟ್ ಡಿಪ್ಲಾಯ್ಡ್ ಲಾರ್ವಾ leads to the queen and worker bee, but the queen always feeds on royal jelly and it becomes fertilized, but which doesn't feed on the royal jelly, only feeds on bee bread, becomes sterile female, that is called worker bee. So this is how we can see the social organization in honeybee also, but it is some other thing. But the sex determination in the honeybee, especially the order of Hymenoptera insects, is this type haploid and diploid mechanism of sex determination. Whereas in the genic theory we can see about how many number of chromosomes and based on their content, so whether they are having the 0.5 or 1.5, especially we can see this mechanism in the genic balance theory is in the Drosophila mechanism also. Okay, so this is all about the allosomic and then haploid diploid mechanism of sex determination, and this is all about the sex determination types. In the next coming up videos we will come across with the human genetics, like the eugenics, where we can see the pedigree charts and pedigree analysis, and then we come across with some of the chromosomal aberrations like the mutations, and then we will come across with some other disorders of the Mendelian and chromosomal disorders. Thank you.